Hello, biology students. This is our second day of the review for our Chapter 12 and 13 tests, which will cover DNA, RNA, and protein synthesis. And so this is off of the study guide that I gave you. I want to point out that there are going to be some short answer questions. There are going to be four. I have indicated the short answer questions on this board with a star. And so when you see a star next to the thing, then you'll know that this is one of the four short answers that will be on the test. And so remember, I will email this home to you, and then you'll have until the next day at 9 a.m. to then make sure to take a picture and then send it off to me. All right, and so let us begin with our DNA. And so we're going to start over here. All right, transformation is the process in which one strain of bacteria is changed into another. And Griffith, therefore, was the discoverer of transformation in bacteria. Avery discovered that DNA was the hereditary material that allowed for transformation. Then we had two scientists working together, Hershey and Chase. They were able to use radioactive isotopes. They used radioactive phosphorus for the DNA, the radioactive sulfur for protein. They infected the bacteria with these viruses. They found out that most of the viruses were contaminated with the phosphorus that was labeled, and therefore again proved that it was the DNA and not the RNA that was the hereditary material. Nucleotides have three parts. They consist of a sugar, a phosphate group, and a nitrogenous base. Remember that Chargaff's rules, apples are in trees, cars are in garages, and so A matches up with T, and C matches up with G. Wilkins and Franklin were British scientists who both used X-ray crystallography to determine what the DNA structure is, and so based upon the pictures that they took, Watson and Crick developed what is now the famous double helix model of DNA. All right, moving on, we're going to now look at chromosomes and DNA replication. In prokaryotes, we're going to find that the DNA is located in the cytoplasm since there's no nucleus. In eukaryotes, then the DNA is found, in fact, in the nucleus. In prokaryotes, the DNA is one circular chromosome, whereas in eukaryotes, it is in a set of chromosomes. And so remember, again, for humans, we have 23 pairs, so a total of 46 chromosomes. Looking at the specific enzymes involved with DNA replication, we have the enzyme helicase, which will separate out the hydrogen bonds of DNA. We have the single-strand binding proteins, which are going to abbreviate as SSBPs. This prevents the reannealing or the reattachment of the DNA once the helicase is unwounded. We also have the principal enzyme in this process called DNA polymerase. Its job is to add complementary nucleotides to the original DNA strands. And then we also have an enzyme called ligase, which will join together the Okazaki fragments. Here's one of the short answer questions. DNA replication is semi-conservative because it contains one parental strand and one new strand. All right. The replication fork is the site where DNA replication occurs. The leading strand is the strand of DNA that is continuously synthesized in one long piece. The lagging strand, as the name might suggest, lags behind the leading strand, and it's made up of Okazaki fragments. All right. Next, we're going to look at the three differences of DNA and RNA. First of all, the sugar in DNA is deoxyribose, hence the D in DNA. The sugar in RNA is ribose, which is why it has the R in RNA. A second difference is in the bases. Remember that DNA contains thymine, or T. RNA contains uracil, or U. DNA is double-stranded, and RNA is single-stranded. So these are the three differences between the different types of nucleic acids. All right. We can also now talk about the three types of RNA and their functions. First of all, we have messenger RNA, which we're going to abbreviate as mRNA. This carries copies of instructions for making proteins. We also have ribosomal RNA, little r RNA. This is used to make ribosomes, which are then involved themselves in making proteins. Finally, we have tRNA, which stands for transfer RNA. They transfer amino acids to the ribosome based upon the codons in the mRNA. The process of transcription takes DNA and transcribes or converts it to messenger RNA. The main enzyme in this transcription process is RNA polymerase. Introns are not involved in protein coding, whereas exons are involved. All right. A codon, as you'll recall, are 3 consecutive nucleotides that will specify for a specific amino acid. We are able to use the genetic code then to determine what that specific amino acid is. So based upon that protein synthesis worksheet that I gave you guys to do last couple weeks ago. All right. Translation is the other major process that takes the mRNA and therefore converts it into protein. All right. We're going to now turn our attention to mutations. We have different types of mutations. In fact, we have 3. We have, first of all, point mutations. And so a point mutation, as you'll remember, is a gene mutation involving changes in one or more nucleotides. We have substitutions, where we change one base out for another. And then we have insertions, where we're adding in a base, deletions, where we're removing a base. And so remember that the insertions and deletions make up what is known as a frame shift mutation. And so frame shift mutations, notice again we have a star here and here, so these would be 2 of your other short answer problems. I also wanted to point out, because I skipped over it, this is another short answer problem, so let me go back just for a second. Anticodons on tRNAs will bind to codons on the mRNA, and the amino acids from the tRNAs are then used to make proteins. And so this is one of the short answer questions. This is a short answer question, and this is the 4th and final short answer question. So frame shift mutations are called such because they do in fact shift the so-called reading frame, and as you can suspect, frame shift mutations are more dangerous than point mutations. All right, the 4 types of chromosomal mutations, and so I'll give you a picture of these 4 types on the test, and you'll need to identify what they are. We have deletion, we have duplication, we have inversion, and we have translocation. So I'll have you guys refer to the notes for that. Finally, we're going to talk a little bit about gene regulation. We have an operon, which again is a group of genes that operate together to either turn on or off a specific gene. And so the example that we used in class was the LAC operon, which allows for the movement of lactose into a cell membrane. We also have a region called the operator. This is where a repressor will bind to, and it will prevent the transcription of genes. So for example, if there's no lactose present for the LAC operon, there will be a repressor that will bind into the operator sequence and prevent the RNA polymerase from transcribing the DNA into RNA. Finally, we have Hox genes. These set of genes will be important because they control the differentiation of cells and tissues in the embryo. And so if you want to go ahead over the Kahoot from yesterday, if you want to look over what I've talked about here today, and therefore you guys will be then well set then for this chapter 12 and 13 test. And so with that, I hope you all have